Okay, we are all set now. I figured it out. All right, so we were going to finish our Turbo Lab, and if you remember, the Turbo Lab is basically this long, drawn-out semester project that we had where we collected our specimen, we extracted the DNA, we did PCR, and remember, if you haven't put those on a resume, then I would highly recommend doing so. Um, we did the gel electrophoresis, and now we have sequenced it. So we actually have our sequences, and we're in this last phase where we're going to build this phylogenetic tree. And this is not a cell molecular class, so I'm not going to go super deep into it, but here is how we are determining our DNA sequences. Basically, in your DNA molecule, you have a unique code that can identify species, right? And we took one gene. We looked at the RBCL gene, and in those genes, for all our bugs, have various letters that make up this DNA code. And so, obviously, they're super small, and this is stuff that we can't just see like under just a normal microscope. And so, what we have done is we've made billions and billions of copies of our DNA through the processes that we ran in lab. And during that process, um, in order to determine the DNA sequences, the company that is going to sequence it basically puts little fluorescent tags on the end of these DNA segments. One tag for every single letter in the sequence. And when there's a tag placed on that letter, it stops the sequencing, it stops the PCR of those DNA segments. And so what you get are individual length DNA pieces that have this fluorescent tag on them. So for instance, um, you'll have a green tag on A's, a red tag on C's, and a blue tag on C's, and then a black tag on G's. And so what happens is, if you remember, um, through gel electrophoresis, the shorter fragments go first. And so what happens is there's a machine with a laser on the end of it, a color detector, that's just a gel electrophoresis machine. And the pieces, all of the DNA pieces that have been made with their special colored tags get sent out. The shortest ones come out first, and so the laser detects the shortest ones first. And so in sequence, it detects the varying colors, which are then associated with a particular letter. That letter, what it does is it says, oh, at the first spot there was, and so for this, there was a black tag. And so that would tell you, oh, there's G. There was a blue tag at the second letter. Oh, that's a T. There was a red tag at the third letter. That's a C. So that's how they've determined the sequences. And as we know, through um, through sequences, we can determine evolutionary relationships and build phylogenetic trees. And that's what we're going to do today, or I'm going to do for you. Um, so here's how it works basically. If you just look at this little graph here, there's a common ancestor, GCCA, right? Whatever. It shares a relationship by this sequence with all of these bugs, but some are more different than others. And so the further away that you are, the more different that sequence will be. That is sort of the very basic premise of phylogeny if you have forgotten, right? Okay, so I'm going to do it for you right now. That was it. Look at all this happy stuff. Look at all this happy stuff. It's just happy times, really happy times. Okay, I have got it already set up. Create a project, blue line. This is Tuesday at 3 p.m. Class data. So this is what you guys would have done, but I am going to upload. Here's all of your stuff. If you only see one number, so some of you only had one number, like 52, 53, 54, 55, 56. Oh, wow, that's a lot. Is that right? I'm going to double check to make sure that is correct. Okay, that is correct, which is very interesting. Anyway, so these are all of our species. And what I'm going to do is plop them into this really, really cool, oh, wow, okay. Really, really neat resource called DNA Subway, and it's going to build a phylogenetic tree for us using the class data. Okay, so there's a little, if you're number 56, don't worry about that. Okay. So there's a series of steps that we need to do in order to prepare it for the phylogenetic tree. And here I am simply parroting it. If you see the F um, and the F is for forward and the R is for reverse. So we go forward and reverse. Ones with two, we will pair. Basically, the ones that have multiple have a little more um, specificity around whether or not the actual letter that they're going to be looking at is correct. So these one, two, three, four, five. I don't know why there's so many. It's very unusual. Okay, this is good. And then we're gonna go next up. All right, let's do all of them now. And basically, what this does is this will, and I believe this takes a little moment, so I'm gonna pause the recording until it works. Okay, we are back. So what the blast does, and we got a positive hit on every one of them, which is super cool. What it does is it actually tells you what your species is. So here, I'll just run through quickly all of them. This, if I get my thing out of the way. So this, just so that you all can see it. This is 52. 52. So there's a bunch of results here. You had this snail. What I'm going to do is just go quickly. If you want to see your species, then you can pause the video. Here's 53. 53. Ooh, I don't know what yours is. There it is, though, right there. That is the scientific name. I'm not sure what that is for 53, but you can, well, let's check it out. Oh, it's a clam. Wow. It's a little fingernail clam. Oh, so you did have a clam. Wow. All right, cool. 54. You have this weird looking bug. I don't know what this is, but you have a weird looking bug. 55. This is definite. And so I just wanted to point out here, you see that there's one, they're all the same here. And so here are the bit score. The higher the bit score, the more certain that the species they have matched um, is more likely. So like this match is more likely than this match because this bit score is lower. The E stands for the chance that it was that the DNA sequence is matched by chance, and then this shows you how many actual mismatches in that entire sequence there were. So here, another snail, very cool. And I think if you remember that lab where we collected these species, that a lot of you did pick snails because they were big. Another snail, which should be interesting. Ooh, something different. Oh, a damselfly. Somebody picked a damselfly. There it is. Very cool. Many different species. 58, 59. Ooh, a fly. Some of you picked a midge. Very good. You found a fly. Cool. And 60, last but not least. Oh, very cool. This beetle. The dingy diving beetle. I don't know if you guys remember the diving beetles. Dingy diver. So, okay, so there's all of our species. Now what we're going to do is we're going to use that, which it looks like all of those are correct based off my memory, which is good. That means also congratulations, pat yourself on the back, because that means that you have done it correctly. So that's kind of neat. Um, all right, so now what we're going to do is we're going to look at how they are related. So if you're looking at this map here on the screen, you'll see that there's all these tick bars, and then there's a consensus bar, which is right up here. The consensus bar shows each spot on those on that sequence. Um, and what it does is it builds a consensus. So, for instance, I'm going to look at this T right here. All of the dots for all of the specimens that have a dot, the T is representative. So it means that the T was most common in this location, but two species, 58 and 59, had an A. And so they were different than the consensus. This is how the phylogenetic tree is built. So here, same thing. Here's a G. All of these species had a G at this location. All of them shared the same letter, right? And so what they do is they're building these differences to look at who has the most differences and who has the least differences. And this is how they can build their phylogenetic tree. So there's two to build, and I'll just show you both of them. Here is your phylogenetic tree. And so what you'll find is here is our common ancestor back here at this location. And what you'll find is 53 is closely related to 52 and 57, but 52 and 57 are much, much more closely related. So if we go and look at 53, if you can remember, what would be nice is to change the names of them. That's what I was going to have you guys do in class, but here we are. So 53, we looked up 53, was a clam, and that makes total sense because there was no other clams there, right? There was no other clams. So it is by itself. It was very unique, which obviously it was very unique. And then 52 and 57, I think, as I recall, were both the same exact species, which is this pond snail. So that's how it works. It's just very, very simple. It's using these differences to build evolutionary relationships. And this one, the NJ phylogeny that it will build, will actually show you what are called bootstrap values. And this is how certain they are about the relationship. And so basically, anywhere you see 100 means that 100% of the trees that were made in this system. So what the system does, if yeah, you guys don't know, what the system does is it makes like thousands and thousands of different possibilities based on the data. And then it says, of those 1,000 trees that were made, what percentage of them had M50, or number 53 by itself up here? And the answer is 100%. So this evolutionary branch is very, very likely. Same thing. These two are very closely related. 100% of the models had them as related, very, very closely. Same thing with 55. And so those are really, really certain relationships. However, if you see, there's one node here that shows this branch is only about 60% of the model had this branch separating 54 and 60, while 100% of them had them very closely related together. Where that branch occurred is not very high in terms of certainty. So this branch here in this phylogenetic tree may be real, it may not be real. We're not really sure. All of these are just estimates. And so pretty interesting. Pretty interesting. But overall, I think our class did pretty good. This is pretty good results. A lot of certainty in these values and shows different relationships. So that's pretty much it. That's what we were going to do today. I'm going to force you to watch this video. Hope you enjoy it. The big announcements. Number one is course evals. I need you to fill out course evals. That would be like super duper helpful. You're not required to anymore, but I use them and value them like really, really incredibly. So your feedback means like a lot to me. It helps me improve the class for future generations, and so I would really, really appreciate it. And then lastly, I didn't get to say goodbye to everyone, but it was a really great class. This was an awesome lab. Lab can be kind of a bummer sometimes because it's 3 hours and late in the day, and I get that, but I really enjoyed this section. And so I just want to say thank you for a great class. And if I don't see you next semester, hopefully I do, but if I don't, good luck with your future endeavors. And if you need anything, feel free to reach out. Okay? Bye, everyone.